Yes. Hello, everyone. Today, I would like to discuss with you all the role of a very important histone H3 core residue, that is H3K56. During the course of our studies with this residue, we found that acetylation of H3K56 aids chromatin accessibility during UV-induced nucleotide excision repair in Saccharomyces cerevisiae. Now, before I talk about my work, I would like to briefly discuss the architecture of eukaryotic genome. So, as
Sorry, Ms. Preeti Khan. Hello.
yes,
Your voice is fluctuating. Like we are not able to hear you properly. Can you please?
am I audible now?
Yes, it is good. It's good. Please
Okay.
carry on. Yeah.
Yes. So, as I was saying that our DNA is packaged into higher order structure called chromatin. The basic structural unit of chromatin is the nucleosome, which consists of 147 base pair of DNA wrapped around an octamer of the four core histones, namely H2A, H2B, H3, and H4. Each of these histones is a very small basic protein with an histone tail and a globular core domain. So, post-translational modification of histone residues has been shown to play a very important role during processes such as DNA replication and transcription. However, our genome is continuously being challenged by several exogenous as well as endogenous agents that lead to DNA damage and which, if not repaired on time, can cause mutation and eventually disease. One of the most potent DNA damaging agents is UV radiations, more specifically UVA and UVB rays, which can penetrate the Earth's atmosphere and have now become the leading cause of skin cancer. So, in response to UV irradiation, two types of lesions are formed in DNA. They are the cyclobutane pyrimidine dimers and the 6,4 photoproducts. Both of these lesions distort the double helical structure of DNA, producing kinks and thereby impeding the process of replication and transcription. As I just mentioned that our chromatin or DNA is packaged into a chromatin structure, while this packaging is very important for maintaining the stability of the genome, however, packaged chromatin is refractory to binding by trans factors that need to access the DNA during any DNA metabolic process. Therefore, during DNA replication, DNA repair, and transcription, dynamic nucleosome rearrangement is required to make the chromatin accessible. This is aided by several factors, which include histone post-translational modifications, chromatin remodelers, histone variants, and histone chaperones. Amongst these factors, histone post-translational modifications play a very critical role. I will be talking about that a little later. So, in placental mammals and in humans, in response to UV-induced DNA damage, the only pathway that is activated is nucleotide excision repair. So NER is the only pathway for removal of UV-induced DNA damage. During NER, large multiprotein complexes participate in the process of damage recognition, excision of the damage, and finally restoration of the chromatin to pre-damage state. So for the large repair complexes to be able to access the site of damage within a chromatin environment, almost 100 base pair of nucleosome-free DNA is required. So simplistically speaking, chromatin accessibility is the rate-limiting step for successful NER. As I mentioned that chromatin accessibility is regulated by histone post-translational modifications primarily, I have listed here some of the very important histone modifications that occur. Amongst these modifications, acetylation of histone residues is the most influential or universal post-translational modification. This is because acetylation of histone residues neutralizes their basic charge, thereby reducing their affinity for DNA and in turn influencing all chromatin-based processes such as chromatin relaxation compaction, interaction of histones with regulatory proteins, as well as nucleosome assembly, disassembly, and DNA wrapping, unwrapping dynamics. Now, I would like to take a quick recap on the structure of histones. Histones possess two structurally distinct domains, that is the histone tails and the globular core domains. As you can see in this figure, both residues within the histone tail and histone core domains are susceptible to acetylation. But because the histone tails protrude out from the histone octamer, previously it was thought that only histone tails could be acetylated to regulate the chromatin structure. However, if you look at the panel on the left, histone tail acetylations can primarily influence decompaction of higher order chromatin structure. However, histone tails cannot regulate nucleosome stability or the accessibility of nucleosomal DNA. It is only the residues on the surface of the histone octamer, that is the globular core residues, whose acetylation can directly influence nucleosome DNA unwrapping. Previous studies have implicated the importance of unwrapping nucleosomal DNA for exposure of the damage. So we chose to understand the role of a very important histone H3 core residue, H3K56. The importance of this residue lies in its location within the nucleosome. That is, it is located at the DNA entry exit site, or the site where the nucleosomal DNA enters or leaves the histone octamer. So acetylation of H3K56 weakens the interaction between the histone DNA ends and the histone octamer, and thereby critically influences nucleosomal DNA unwrapping. Acetylation of K56 has been previously implicated in the process of transcription as well as DNA damage response in mammals. However, sporadic information existed until now on the role of this residue during NER. So, normally in eukaryotic cells, acetylation and deacetylation of histone lysine residues is catalyzed by two specific enzymes, the HATs or the histone acyl transferases and the HDACs. However, in this study, we wanted to understand the role of K56 acetylation. So we mutated the lysine at position 56 of H3 to either arginine, which locks the residue to its constitutively deacetylated state, or we mutated the lysine at position 56 of H3 to glutamine, which locks the residue to the constitutively acetylated state. With the wild type and H3K56 mutant strains, we first checked UV sensitivity. For this, the cells were with or without UV radiation were serially diluted, and you can see here that at 100 joule and 150 joule per meter square, which are the higher doses of UV, in H3K56R cells, that is cells in which K56 residue cannot be acetylated, shows significantly higher levels of UV sensitivity compared to wild type and H3K56Q cells. We also monitored the cell survival rate per dose of UV, and the data clearly revealed that in K56R cells, the cell survival rate was severely impaired compared to wild type and H3K56Q cells. So this led us to question whether the UV sensitivity of H3K56R cells, where the residue is unable to acetylate itself, was because of an inability to remove UV-induced DNA damage. So one of the most frequent forms of UV-induced DNA damage is CPDs. So we monitored the CPD removal rate. For this, the E cells were UV irradiated at a dose of 100 joule per meter square, and then they were allowed to repair for different time points. The DNA was isolated from each repair time point, and it was either mock treated or it was treated with T4 endonuclease 5, and the control and treated samples were then electrophoresed under denaturing conditions. So before I talk about my data, I would like to explain the principle of this experiment. So in response to UV radiation, bulky helix distorting lesions such as CPDs are formed on DNA. Now T4 endonuclease 5 is a CPD specific enzyme, so it specifically makes a cut on the 5' ends of CPDs, thereby generating multiple single-stranded breaks in DNA. Now, when the nucleus treated sample is run on a denaturing conditions in an alkaline gel, if you look at the gel panel, at 0 minute of repair, the plus indicates that the DNA has been subjected to nuclease 5 digestion, and you can see that there is an intense smear towards the bottom half of the gel. Now, with progression of repair, that is 30, 60, and 120 minutes, you can see that the smear has started to migrate upwards. This is because with progression of repair, the CPDs are being removed. So there will be lesser nuclease cut sites on DNA. So the larger fragments migrate slowly. So the increase in the smear upwards is an indication of CPD removal. So we quantified these bands, and subsequently we monitored the CPD removal, and we saw that in H3K56R cells where the residue cannot be acetylated, there was significantly lower levels of CPD removal compared to wild type. Surprisingly, in H3K56Q cells where the residue is constitutively acetylated, CPD removal was rather promoted, especially at the early time points, that is 30 minutes to 1 hour of repair. So from the sensitivity and the CPD removal data, it was evident that H3K56 acetylation possibly has a role to play during NER. Now, histone acetylations don't not only participate in chromatin accessibility, but also in chromatin restoration post-damage removal. So next we wanted to check during which phase of NER did acetylation of K56 have a role. So for this, we subjected the E cells to UV radiation, and we allowed the cells to repair for varying time points. And we monitored through chip assays with antibodies against K56 acetylation the status of this residue during NER at the 4 east locus, HML and telomere, which represent the transcriptionally silent regions, GAL10, which represents the transcriptionally repressed locus, and RPB2, active locus. And in all of these 4 loci, it was evident that immediately in response to UV, within the first 2 minutes of repair, hyperacetylation of H3K56 occurred. This data clearly revealed that acetylation of H3K56 is required during the early phases of UV-induced NER. Now, I would like to talk about the NER pathway in greater details here. So in eukaryotes, NER, based on the initial damage recognition step, is divided into 2 pathways, the tcNER or transcription coupled NER, which is mainly activated due to a stalled RNA polymerase, and the ggNER, which is the NER pathway responsible for removal of damage all throughout the genome. And 2 proteins which are unique to this pathway are the RAD7 and RAD16 complex. And this protein complex is indispensable for removal of UV-induced DNA damage through the ggNER pathway. Previous studies have reported that in absence of damage, the RAD7 and 16 complex together remains locked to certain regions of the genome with the help of barrier histones. In response to UV-mediated DNA damage, some NER responsive event occurs, which is possibly a histone acetylation and chromatin remodeling that facilitates the removal of this barrier histone and subsequent redistribution of the complex to the site of damage. At the site of damage, the RAD16 complex is responsible for HAT-mediated hyperacetylation of 2 very important histone H3 tail residues, that is H3K9 and H3K14. This generates a platform for recruitment of downstream repair factors and efficient damage removal. So as we saw that H3K56 acetylation occurred very early during NER, within first 2 minutes of repair. So we questioned whether the NER responsive event that promotes RAD16 redistribution to the site of damage was H3K56 acetylation or not. So through chip assays with antibodies against RAD16, we monitored the recruitment of this protein at the 4 east locus during NER. And as you can see, it is very evident that in presence of constitutive acetylation of H3K56, RAD16 recruitment was promoted during NER, and surprisingly, it was also higher compared to wild type, even in the absence of any kind of damage, UV damage. So this clearly indicated that Rad16 redistribution during NER was being, in response to UV, was being regulated by some event which involves H3K56 acetylation. And in H3K56Q, because the residue is constitutively acetylated, so the recruitment of this protein becomes UV independent. So, as I mentioned, that Rad16 protein is responsible for the recruitment and HAT-mediated hyperacetylation of H3K9 and H3K14. We next wanted to check the influence of K56 acetylation on these residues. And as you can see here, the left panel indicates the H3K14 acetylation levels and the right indicates H3K9 acetylation levels. And in both of these histone residues, you can see that in K56Q cells, hyperacetylation of H3K9 and H3K14 occurs immediately in response to UV within 0 minute of repair, and it is higher all throughout repair, especially in the early time points compared to wild type. Additionally, what transpired out from this data is that in H3K56R cells under minus UV conditions, surprisingly, K9 and K14 acetylation levels were rather higher compared to wild type and K56Q. However, it is noteworthy to mention here that despite having higher basal levels of K9 and K14 acetylation, we have earlier seen that in H3K56R cells, CPD removal rate is impaired. So this possibly could be explained by the fact that in H3K56R cells, we see that Rad16 is not recruited in response to UV. So lack of Rad16 recruitment and Rad16 mediated redistribution of K9 and K14 to sites of damage may be one of the possible explanations. So to summarize so far, we can say that in response to UV, H3K56 acetylation occurs, which promotes Rad16 recruitment, which in turn leads to hyperacetylation of H3K9 and H3K14. Now, the next question was, how did this impact the chromatin structure during NER? For this, I performed two assays: an in vivo micrococcal nuclease assay and an in vitro plasmid supercoiling assay. Now, what exactly is a micrococcal nuclease? A micrococcal nuclease is an enzyme. It's basically an endonuclease which is able to produce double-stranded cuts within nucleosome linker regions. The nucleosome linker regions are the linker DNA between two adjacent nucleosomes. So that region of the DNA is accessible. Hence, it is susceptible to MNase digestion. So the efficiency of MNase digestion principally depends on the chromatin accessibility. So higher the accessibility, higher will be the digestion by MNase. Now on the right panel here, you can see the bands. These bands indicate the nucleosomal DNA bands. The nucleosomal DNA bands, here these bands actually represent that portion of the DNA which has been protected from MNase digestion. So we obtained the digestion profiles of wild type K56R and K56Q mutants. Both under, first let us look at the minus UV gel panel. There was clear difference in the digestion profile, more specifically the 10 units of MNase concentration in wild type and K56Q cells. As you can see, that the nucleosomal DNA band intensity in K56Q cells is fuzzier and less distinct compared to wild type cells. We also plotted the densitometric scans of these bands, and as you can see that in wild type and H3K56R cells, the peak heights are higher. Now these peak heights actually represent the intensity of the nucleosomal DNA bands. Each of these bands is represented as a peak here. So the higher the intensity of the nucleosomal DNA bands, greater will be the height of the peak. That means that the DNA is less accessible and less prone to digestion by the enzyme. As you can see in H3K56Q cells, the peak heights are lower, which clearly indicated that under control conditions, K56Q kept the chromatin more accessible. To further our understanding, we obtained MNase digestion profiles during NER. Sorry. And we could see that by 30 minutes of repair, In wild-type cells, perceptible chromatin rearrangements occur, leading to generation of very compact chromatin and fuzzier lower-order structures. Such structure is also evident in H3K56Q cells. However, in H3K56R cells, as you can see, that the chromatin architecture more or less remains unchanged all throughout the repair time points. That is, there is no uh, marked change in the chromatin architecture all throughout repair. This clearly indicated that in wild-type cells, in response to UV, some chromatin regulation occurs, and presence of H3K56 acetylation possibly promotes such chromatin modulations. So, to further our understanding, we performed an in vitro plasmid supercoiling assay. Here, we irradiated the E cells to UV and allowed them to repair for different time points. We produced cell extracts from these repair protein time points, and then the cell extracts were then incubated in presence of plasmid, which was previously relaxed with topoisomerase 1. And the plasmid DNA was then isolated and electrophoresed in alkaline gel. Electrophoresed. So, basically, the principle here is that as DNA wraps around an histone octamer, it generates one negative supercoil per core particle. So you can see that in wild-type cell extracts, under control conditions, the plasmid DNA was primarily kept in the supercoil state. On the other hand, with H3K56Q cell extracts, we saw that the plasmid DNA was more or less primarily in the relaxed state. However, in H3K56R cell extracts, the plasmid was neither fully compact nor was it fully relaxed. Additionally, what transpired from this data is that in wild type cells, during NER, you can see that highly supercoiled conformation moves to a highly relaxed conformation. So there is chromatin modulations occurring even in plasmid DNA, that is in an in vitro assay. However, and most importantly, the plasmid conformation that wild-type cell extracts achieve by 60 minutes of repair is very similar to the conformation of plasmids with K56Q cell extracts, both in presence and absence of UV. Additionally, in case of H3K56R cell extracts, you can see that the plasmid is more or less static and the conformation remains more or less unchanged all throughout repair. So this data again clearly substantiated that H3K56 acetylation essentially regulates the chromatin compaction relaxation and DNA accessibility during NER. Therefore, to summarize the data, in response to UV, wild-type cells achieve a flexible chromatin structure due to an immediate hyperacetylation of H3K56 residue. This leads to the recruitment of the repair factor RAT7, RAT16. This in turn leads to hyperacetylation of histone tail residues H3K9 and H3K14. The histone coat generated by the hyperacetylation of these residues promotes chromatin accessibility and recruitment of the downstream repair factors, consequently leading to efficient damage removal during nucleotide excision repair. Now, I would like to end by saying that histone acetylations have long been correlated with cancer therapy. And currently, a lot of research is being done with HATs and HDAC inhibitors, which are being utilized to block certain repair proteins or DNA damage response pathways in cancer cells to induce cell death. So, because chromatin plasticity is very important during DNA damage response, and from this study, as we could see that K56 acetylation plays a very critical role in chromatin plasticity. So in future, studying the role of this residue in higher eukaryotes and mammals could possibly open up new avenues for future studies in anti-cancer therapy. Lastly, I would like to acknowledge and thank my supervisor, Dr. Ronita Nag Chaudhary, for her unconditional support and guidance, and my fellow lab members for their constant help. I would also like to thank my funding agencies, DST and CSIR India. And last but not the least, I would like to thank St.Xavier's College, Kolkata, India, for providing me with the research facilities. And I would also like to thank the organizers of this webinar for giving me the opportunity to present my work. Thank you so much.